With Doubtnut, get instant video solutions to all your maths, physics, chemistry, and biology doubts. Just click the image of the question, crop the question, and get instant video solution. Download Doubtnut app today.
Hello everyone, this is a question. Let me explain the question to you. Aldopentoses, named as ribose and 2-deoxyribose, are found in nucleic acid. What is their relative configuration? Okay, so we have to basically tell the relative configuration of the aldopentoses that are given to us, right? So aldopentoses given to us, so clearly we are having 5 carbon atoms that would be present in the structure, and it's an aldo is clearly telling us that there would be an aldehyde group also that is present. Okay, so if we convert the configuration to a cyclic ring, we can get a structure something like this. O, a 5-membered ring would be there, which is present like this. Okay, and according to the configuration, the OH at the primary first carbon is present in the upward direction, then H, then OH again in the upward direction. Here at third carbon, OH is downward, and at the fourth, this is the 5th carbon, primary alcohol, and this is the hydrogen. Okay, this is the basic structure for the ribose. The second one is 2-deoxyribose. So what happens in that? The second carbon OH is eliminated. So then we get a 2-deoxyribose, which will look something like this. Again, a 5-membered ring is there. The position at first carbon remains the same, OH, but at second, that OH group from ribose is now removed. That is why we are calling it deoxy, right? And at third, OH in the downward direction, and then this is H, and finally the CH2OH. Right? Now, if we consider the naming according to the structure, we can clearly see that they are the beta form. This is for ribose and this is for 2-deoxyribose, and they all are in the beta configuration. So we can mention that beta ribose and beta 2-deoxyribose. Now we have to check whether they have D or L configuration. For that, we need to remember a small point. In the case of cyclic structures, if we have to decide D or L configuration, then we need to focus on the second last carbon atom. Right, if the second last carbon atom, if at second last carbon atom, the OH group has to be noticed. OH group present from bottom side is to be noticed. So talking in detail of that, what we mean by the second last carbon, like if this is the 5th carbon in the beta ribose, like this is first, second, third, fourth and 5th, then we need to see the second last carbon from the bottom side. So the second last carbon from bottom side, that is 4, 5, 4, 3. That will be this carbon at third position. Here we have to see OH attachment, right? So we can clearly see that in this structure, OH is attached in the downward configuration. So it will be the D configuration. If OH will be in upward, it would be L. So here we can say that it is D beta ribose. Similarly, watching the second structure, this is first, second, third, fourth and 5th carbon. Here also we have to observe the second last carbon from the bottom side, that is again 3. Here as well, OH is attached in the downward direction, so this configuration is also D beta. Okay, so the final answer is D beta ribose and D beta 2-deoxyribose. Okay, thank you.
For class 6 to 12, IIT JEE and NEET level. Trusted by more than 5 crore students. Download Doubtnut app today.